but we're looking now for better tolerance in the mouse and the rat. It's also driven by toxicology now, and we're looking for various high therapeutic index peptides. And we've now got a series of these, and we have been able recently to show that we can get high activity with low effectivity in kidney as well. So we're developing those as drugs. Just about to finish here. So I just want to say that as well as Duchenne muscular dystrophy, which we're now taking towards the clinic in our drug development, we're also working on spinal muscular atrophy. There's a series of papers we've published on this. We've been able to show that our peptides are able to cross the blood-brain barrier, which means that we can deliver by intravenous activity. And we're talking to Biogen, who have recently produced a drug called nusinersen, and we hope to develop this as a second-generation compound. We also work on myotonic dystrophy and got some really good results there. I haven't had time to show you. And we've also very recently delivered into the mitochondria, using PNA into the mitochondria. So there's a number of things that I'm not able to say, but now my lab is closing. Kurt Hogeweis is going back to Belgium to develop this technique of delivery into the mitochondria. We are developing this as a drug. This is my final group here. Andre and Frank are my last two postdocs. I have a collaboration with Matthew Wood. I shall be working very strongly with Matthew Wood to develop these compounds. And we're starting a company in Matthew Wood's lab now to develop these. A company we'll call PepGen, to bring peptide PMO to the clinic. And I just want to thank you now. Many co-workers have passed through my lab, and also to my heritage of Govind Khurana and Tom for setting me off on this trail. And thanks to many recent funders and supporters here who have worked with me. And thank you very much for your time. Sorry, I'm just a little bit over.
Thank you, Mike, for this very interesting work. So most of it is really nuclear targeted, right?
Yes, the nuclear targeting is the most important, and getting things into the nucleus requires a peptide, and it requires an arginine-rich peptide to get there. Targeting microRNA is very different. There you can target without requirements of a peptide, but here it's the peptide delivery, and this is what we're now concentrating on, finding peptides which are short enough and with fewer arginines as possible. We can keep the toxicology down, in kidney in particular is an issue, but yes, sufficiently high enough activity in mouse models. So we're quite excited about this, and we think we can bring these to clinical trials.
Great. One question. Somebody. Yes.
Yeah, it's a very interesting question. We actually don't know how it crosses the blood-brain barrier. Probably the arginine-rich peptides use absorptive transcytosis. We don't think it's a receptor-mediated process, although we're looking at peptides which also have a component involving receptor-mediated. But these we think are not receptor-mediated, and we don't actually know precisely how it gets across the blood-brain barrier, but it certainly gets into the spinal cord and into the brain of these mouse models, and we've shown that in a couple of examples published quite recently with one of our peptides and another peptide. And we're now developing this new class of shorter peptides here. But so we're not. It's very difficult to do experiments in this area, but we think it's an absorptive transcytosis. Sorry? Percentage getting into it's very difficult to assess that. A very small amount. A very, very small.
Okay. He wants a quick question.
Yeah. No, there's no signal. Basically, you get released what happens is you go through an endocytotic pathway taken up by one of the endocytotic mechanisms into the early endosome, then into the multivesicular bodies, late endosome, these things are then released. The arginines, the hydrophobic components, help to distort the membrane, the endosomal membrane, allowing leakage, and then it automatically goes in the nucleus. Correct. They'll perturb the membrane. Yeah. Yeah. You don't need anything to go in the nucleus. It's small enough. If your oligo is small enough, if you have a very long oligo, 50 or 100 long, then you need a transporter system. But these are short enough to just go straight into the nucleus. There's no barrier. Once you get into the cytosol.
Okay, lunchtime. You can talk to me. Okay.
Okay.
All right. Our next speaker is Esteem Ansari. Esteem is not a member of the Karana alum in the strict sense of the terms, but he is an honorary member and most honorable member of the Karana team. Yeah. So Esteem is has done a lot for the Karana program. He started in Wisconsin the Karana internship program that allows lots of students from this country to spend a summer in the United States with the help of the Indian government. So Esteem is just done a wonderful job there, and he's expanding into all kinds of stuff. He might even say a few words about it. But today I think what he's going to say mostly is give a talk on creating synthetic genome switches to control cell fate. Esteem.
So it's an honor and a pleasure to be here. I had never trained with Gobind, but I met him briefly with Tom. And I have read his papers and I took inspiration from his words. So it's the Eklavya model of training. Many of the Indians here will know what that means. From a distance, he trained me to be a good scientist. So what I'm going to tell you about is how that fusion of bringing chemistry to biology has influenced my own work. The talk is in two parts. 2/3 of it will be about making precision therapeutics that Marsha, wherever she is, where's Marsha? Yes, Marsha disparaged. So I'll make a case for it. And the third of the talk will not depend upon the timing. It might go faster than a third. But that'll be about the Khurana program, where we are planning to take it and what the next steps might be, and seeking your advice on how it should be crafted as we go forward. So this is the topic of the day for my talk anyways. Let me start the timer because I saw what Tom did to speakers who are a little longer than they should have been. So I'll talk about transcription factors as we understand them chemically. So people know transcription factors control gene expression. If it moves forward, we'll see. It's not moving. They have to point to there, I see. So these are the three areas that my lab works in. We try to understand mechanisms in model organisms to try and figure out how the machinery actually works. Surprisingly, it's still being determined. We make synthetic transcription factors. I'll tell you more about that. And we also try to make synthetic genomes altogether. So here, we try to understand how signals, just like Marsha was describing in her very elegant work over the years, how signals lead to activation of transcription factors in this case. So she talked about BAC1. This is a more generic version of that idea that there is some molecule protein that recognizes that it is now to go find its place in the genome. Once it gets to that place, it either turns on, which is how we typically think of transcription factors, but also in the example of BAC1 with maths, it turns off. So how you regulate the genome, what genes you target, how you turn it on, how that specificity is defined. Where is this factor supposed to go? We spend a lot of time trying to understand that. Once it gets there, how does it engage the machinery? We spend a lot of time doing that. And most of these studies are done in yeast, bacteria, Drosophila model organisms. And that has influenced how we go about doing this, which is making so similar or artificial transcription factors, or ATFs, that can target sequences that you're interested in. So you now by design have made molecules that can target a specific site in the genome or multiple related sites. And by making those molecules and conferring on it on or off switch capabilities, you can now drive genes. And by driving specific genes and their networks, you can drive cell fate. right? So that's a part of what we do. We've taken cells that are fibroblasts, converted them to stem cells. We've taken stem cells, converted them to heart, neuron, pancreas, etc. Beta pancreatic cells. So that's one part of my lab that's focused on. Beyond this, this is protein-based. You've changed the tips of this protein to target a specific site in the genome. You've changed regulatory elements to turn them on or off, proximal genes on or off. We've taken the next step to make these things completely synthetic. But let me just finish the idea. Beyond transcription factors, so these gene switches that can target sites in the genome, turn them on, off in a controlled way, can you make an entire new genome? Right? So you heard some really elegant work described by Dr. Gate right now. And yes, there you are. And Mark-Ruder's chemistry, using that exact same chemistry with a photoprotected group and that projector, in fact, the digital micro mirrors that are in that projector, you can on a glass slide synthesize 6 million different oligonucleotides, 100 nucleotides long. You can make genome scale genomes in a matter of a night. And then we have attached to that microfluidic devices where you can collect similarly sequenced genes, sequences, separate them out, assemble those things into larger fragments, make things up to 16 to 600,000 base pairs in a matter of a week. Completely clean. So that's where the world is going. So you heard about a gene being made in 3 months. You can make a genome in a day or two. If you're at one of these big places, in my lab, you can do it in a week. So that is where we are in my lab on the whole. But this is what I want to tell you about today. So this is truly bringing chemistry to biology. And I thought this would be the audience to tell the story. And this is, of course, inspired by the way Gobind did chemistry. Long before Stuart Schreiber named chemical biology, I used to tell him there's this book by a man called Gobind Khurana. He's just down the street. And he used to smile and say, but Esteem, this is different. I don't think so. My time's up already? How much time have you given me? But basically it comes back to the ideas that are borrowed from biology. Right? So people have been looking at natural transcription factors for a very long time, since the 60s. And what they've determined, people with whom I trained, have determined that basically you need 2 bits of information. One bit of information is a DVD, DNA binding domain. It'll take you where you need to go in the genome. So it's conferring specificity as to location on the genome. The second bit of information you need is the interaction domain. At least I call it an interaction domain. Other people call it activation domains, repression domains, effector domains, etc. Multiple names. But the idea is that you're going to bring in the machinery to do something interesting. So if you have those 2 together, you should be able to turn on a gene. And what we've done is we've taken this thought and made synthetic counterparts. No protein here. It's a completely synthetic counterpart that has those 2 bits of information. And we've taken that information and targeted it to a disease called Friedreich's ataxia. This happens to people, leads to scoliosis, then leads to shutdown of the heart, and then loss of cerebellar material, so people can't walk very well, cannot control their motor muscles and neurons. And so basically you have ataxia. Ataxia means you cannot walk, you have trouble walking. Ataxia of this kind actually emerges because a gene called frataxin right here is not being synthesized. We talked about mitochondrial genes. This is a nuclear encoded protein that then goes to the mitochondria and functions there in electron transfer chain, doing the iron sulfur clusters that are required by the electron transfer chain. So there's a connectivity to what Marsha was just talking about. So how does this happen? Why does frataxin go down? So the normal gene, frataxin gene, has 5 exons, and it has between the first and the second exon a GAA repeat, a trinucleotide repeat, typically under 30. Everybody sitting in this room has at least 1 allele, meaning one of the 2 genes that you have from your father or your mother, that has less than 30 repeats. If you have more than 30 repeats, this is what happens. Your transcription levels, your levels of FXN RNA, frataxin RNA go down, and you develop Friedreich's ataxia. Depending on the number of repeats you have, it happens sooner or later. And you have to have both alleles hit in this way. If you have one, you can be a carrier and you'll be just fine. Make half as much protein, you're fine. However, people have been studying this disease because it's one of the most prominent recessive ataxias that are inherited. It's a monogenic disease, meaning one gene leads to this disease. So if you can fix this gene, you'll fix the disease. Along the same lines as Dr. Gates' arguments of muscular dystrophy, etc. The understanding of how this happens might lead to a rational way of tackling it. And so people have studied what this GAA repeat does. So now the GAA repeat, on one allele you have 650 copies of it, on the other one you have 1,000 copies of it. And this particular person, that's his brother, you could easily watch how that lack of that gene affected the motor functions, then affected the heart, and the brother eventually died, but the cells were retained, and we've been working on these cells. And so what people have done is actually try to see compare these two, what is happening here? Why is it that you're not making this RNA? So here's one model, a lot of very beautiful work done here in Delhi and in Chandigarh, led to the argument that this sequence can form unusual DNA. It can form triplexes, it can form R loops, it can form sticky DNA. And the polymerase, this is the RNA polymerase that is transcribing that gene, gets stuck. It's trapped in that state. The other models that have existed out there, have become more popular recently, are more driven by chromatin, so that this sequence here somehow leads to wrapping up of this DNA in a repressive state. So DNA is wrapped around nucleosomes. Those nucleosomes are marked with repressive marks. Those repressive marks are recognized by proteins, and the DNA is condensed. And now polymerase has trouble getting through. Both these groups, and they sort of argue that perhaps this leads to that, but there's no clarity in terms of which one of these mechanisms is blocking transcription. But the evidence is clear that transcription in fact does go down, and a lot of people have validated that that is true. So what we decided to ask is, at what level are those repeats inhibiting transcription, right? There are some people who argue that polymerase can't load at all, and so what we did is a simple chip experiment. This is where you throw in formaldehyde, you cross-link the protein to DNA, you pull out the protein of interest, in this case RNA polymerase that is going to transcribe that gene, and you ask, is it there at all, first of all? Second, if it's there, where is it? Is it at the promoter? Here's where the gene is starting, and the answer is not quite. It's left the station. It's made the first exon. Is it at the repeats? Is it trapped in the repeats, as the model for sticky DNA suggested? Not quite. It's not there at all. In fact, it stops well before it hits the repeats. All right? So we know that there's a stalled polymerase there, and it has trouble getting through the repeats. And the work was done by Matt Lawler at Harvard Medical School and my students. So the idea is, the simple idea that we decided to pursue, is that here are those GAA repeats. They are doing something to shut this polymerase down, prevent it from going through, but they do not prevent its engagement at the promoter and initiation of transcription. So it starts just fine, hits the end of the first exon, and then has trouble getting through. The idea is that, can we make a molecule that can dock into these GAA repeats in that way? So this part of it will recognize GAA and deliver an interaction domain, through an interaction domain, the elongation machinery, something that will allow the polymerase to get through this repressive region. Okay? And so if it does, so one half of the molecule is going to recognize, this part is going to recognize GAA, the other part is going to deliver the elongation machinery, and that is going to act on polymerase to mark it in a way that it can collect friends and family that help it along through the repressive region, come out on the other side, and follow all the necessary cues that are in that gene to do what it needs to do: splice, terminate, and then let the transcript be exported and converted into protein. So you have this two-modular molecule that we are going to make, bifunctional molecule. One module has to be DNA targeting. These are the molecules that are currently used. These are the strategies that are currently used commonly to target sequences in the genome. The CRISPR-Cas approach is incredibly easy to use, intuitive. Even high school students do this easily. So this has become quite popular very rapidly. This perhaps is the more stringent way of targeting the genome. These are proteins that can be designed to target single nucleotides, and they are very precise, much more precise than this or anything else. Zinc fingers used to be the way people used to target the genome. There are many complications because of fingers talking to each other and influencing binding. And then there are these small molecules that are derived from distamycin, versions of distamycin, that can sit in the minor groove, not the major groove, sit in the minor groove and drive and interfere with transcription, but they can be modified in a way to target specific sequences. So this is the size of the molecule, and this is, if you were to draw the chemical structure, what the molecule looks like. So Peter Derwin, Lown, Wemmer, a lot of people spent many years trying to understand how distamycin, which is just these three N-methylperols attached with amide bonds, bind DNA. And it turns out that this sequence is made by bacteria, and it targets AT-rich sequences, and it keeps bacteria with AT-rich genomes from being replicated, from growing, sort of holds them. It's a chemical warfare, if you will. They're preventing their enemies or neighbors, if you will. Typically it's the neighbors, isn't it? Who are your worst enemies? So they tell me. In any case, so what Derwin and others have done is replace these rings with other types of rings. So here you have a hydroxymethylperol, you have N-methylimidazole, and these are just some of the examples. Many, many rings have been used because it's such a modular system with a peptide bond, with an amide bond right there. What they've also done, people who've been developing this field over 20 years, is attach two molecules together with a flexible tether, and when you stack them, they fit in the minor groove quite snugly, and when they are fit that way, they tend to be very specific. A stack of N-methylimidazole and an N-methylperol almost always takes you to a G base pair. If you flip the orientation, you target a CG base pair. If you have a perol against a perol, where is it? There it is. You cannot tell the difference between A and T, but you can certainly tell the difference between a C and G. So these sorts of modular recognition rules that were developed by Derwin and colleagues, many colleagues in fact, have been codified in this manner, and we use that to figure out how specific are these molecules. We made a variety of molecules, we tested them against single sequences, then we realized that that may be misleading because they may bind other things we just don't know. So we made microarrays that had millions of sequences on them, every possible 12mer represented on the array as a duplex, so that it's an unbiased screen of what the molecule likes. You label the molecule, you put it on the array, etc., etc. 10 years of my life was spent doing that sort of thing, right? And in the end, my students said, but how does it work in a cell? And that's completely changed how we think about this. So here, Graham Irwin, who is now at Stanford, basically did the simple experiment that we do with proteins all the time. You have a cross-linker, you cross-link proteins to DNA, just like I showed you for polymerase, to ask where is the polymerase? It's past the start site, you can see it very clearly. So you ask, where is the small molecule? Now it turns out, unfortunately, and we could have predicted this just from looking at the chemical structure, that formaldehyde is not going to do it for us. Now my colleague at Caltech, Peter Derwin, soldiered on. He attached various side chains to ensure that perhaps you could just use the same approach, formaldehyde cross-link, come in with click chemistry, pull out the compound and the DNA that is cross-linked to that compound. And they spent a lot of time. What Graham did was a little less elegant. He decided to go get sorolin and a biotin. So biotin is a affinity tag, if you will, that is quite robust, 10 to the 15th, if I'm not mistaken, KD. And sorolin is a planar molecule that intercalates between AT base pairs and makes a photoadduct depending on the wavelength that you shoot it with, di or mono. A bulky molecule. He made sure that there were linkers so that the cross-linking module could go find wherever it needed to dock. We did the calculations, what is the frequency of AT sequences in the genome, etc. Based on that, we came up with this model. And this is very different from what my colleagues in the chemical world have been focused on. The idea is that here's that small molecule that I've been showing you. Here's a crystal structure actually of that small molecule bound to the minor groove, bound to DNA that is wrapped around the nucleosome by the person who did the structure of nucleosome, Carolyn Luger. So she did these structures, and you can clearly see the molecule can bind nucleosomal DNA. But what we did not expect, that it would actually bind clusters of these sites. So there are multiple sites, and red means strong binding, really bad binding, sort of exemplified here, that the molecules can bind as a cluster, and that is where you really find them. If you have a single strong binding site, and that has been the, the brass ring, the challenge that everybody has chased, can I make a molecule that will target one site in the genome? And this is perhaps the lesson. Looking at how biology does it, the one thing that we do, we try and see how transcription factors do it. And looking at the way, people think about it from chemical perspective, like trying to dock something into an active site, a unique location where a molecule can be fitted just so. Those two ideas don't quite fit when you're faced with the genome. You have many local minima, and the way natural factors do it, that they sample the genome, and they tend to stick around where there are other proteins that stabilize them. There's a couple equilibria that holds them there. As the dwell time increases, these things can do their biological function from these locations. There isn't one transcription factor that functions only at one site, ever. All right? And so we brought that biological thinking back, and that data was telling us that our molecules were doing exactly that. And so we started using this idea, which we call summation of sites, to go look through the genome to see where these molecules would go. And what we found, of course, we came back to this Friedreich's ataxia model, because what you have here is clusters of GAA sequences. So we made molecules that will target those GAA sequences. We attach to it a molecule that will dock into a specific protein that brings in the elongation machinery that will allow polymerase to get through. So here is the DNA binding module. Here is the interaction module. The interaction module actually looks like this. It's a molecule called JQ1 made by a postdoc of an undergraduate of mine, who's now the head of Novartis. So JQ1 is Junchi1. Junchi1's molecule allows us to conjugate at that end a PEG linker attached to our polyamide. And basically, the length of the PEG linker is such that it has to permit the binding of these two modules into their respective biomolecules, the nucleosome and BRD4, BD1 domain. These are the molecules that we made. Here's the linear architecture polyamide. This is a hairpin. This is a linear molecule that targets GAA, GAA, GAA. All right, so that's the representation of that. Imidazoles are colored in. And so you have just the plain imidazole, or you have, after a certain linker, here's the PEG linker that we used, is JQ1. So in any case, now we have this conjugate, which we call SYNTEF1. And we have another one that targets a different sequence. There's the HIF1 alpha targeting site, also attached to JQ1. And so here's this story just appeared 2 days ago in Science. And so here's the test. Here's the test where we now take the molecule and we ask, where does the molecule go? The molecule, in fact, does go to those GAA sites, the repeats in that particular cell line. We ask then, once it gets there, does it bring in BRD4? The answer is yes, it does bring in BRD4. Here you are. This is cell treated with the polyamide alone. This is a cell treated with the polyamide alone. And this is a cell treated and JQ1 independently. They are not conjugated. But when you conjugate them, now you can very clearly see that BRD4 is being brought in. This elongation module is being brought in right over those repeats. And it seems to be interacting with something upstream where there is no compound. So we argue that perhaps this elongation machine is talking to the stalled polymerase through other partners. And there's evidence for that there are in fact proteins that mediate that interaction. And you can see that interaction then leaves polymerase. Here's polymerase. That was BRD4. Polymerase now escapes from that site and goes into the gene. And what is even more compelling than the little bit of polymerase that you see get into the gene is that when it does that, associated with BRD4 is a kinase that is critical for elongation called positive transcription elongation factor B, PTEF-B. So this thing here phosphorylates the polymerase on its tail, and that phosphorylation is absolutely important. It's a mark, it's a signal that, okay, now bring the machine that will allow me to get through this part. And that mark is unmistakable, right? So when you have the compound, here's the repeat, here's that little bit of polymerase, but it is modified in a way that it should be modified. And what is even more gratifying is that as the molecule, as the polymerase transcribes right past these repeats and into the gene and then all the way to the stop sign, you find that there's another peak there exactly where it should be. And our molecule is not working at that distance, but the polymerase is using the cues in the gene to do what it needs to do to make the functional transcript. So here's the functional transcript being made in a diseased cell. Here's the functional transcript not being perturbed significantly in a healthy cell, right? So you can see that as you give more compound, you make more restore function to almost 100%. You see that the other molecule does not do this. And not only that, when you look at the entire transcriptome, all RNAs in the cell, in this disease cell, the one thing that is being moved significantly and maximally is the gene that we are targeting. So it's not affecting a lot of different things. And in this case, where there's a healthy cell, the same compound is doing virtually nothing in this volcano plot. You make the RNA, you splice it, you make the protein, you process it to the size it needs to be, and you do not perturb anything in a normal cell. So we have a molecule that can now reverse the problems that you see with frataxin. The question is now, does it do anything beyond that? It is a small molecule, and I just told you earlier that it samples the genome in many places and sticks around in many places to different levels. Right? The dwell time at different locations differs depending on the affinity and number of binding sites. And so here we are ranking them by order, 250 locations in the genome where this compound can go. So it's sampling everything. It stays a little longer at the frataxin gene, but it stays long enough to bring in the partner, BRD4, to those sites. And yet what we're seeing is a single gene is moving. If there are 250, let's assume that this is at least half of them. There should be 125 genes that have moved to some extent. And when you draw these lines, there are only 11 genes that move. Half of them are in response to the fact that you've restored frataxin function. Okay, the other half, you don't know what they are. But in any case, when you look a little closer, this is what you see.